right, so today we are going to talk about hemostasis, which is blood clotting. Okay, so there are three processes involved. They are the blood vessel spasm, platelet plug formation, and the blood clot formation. So the vascular spasm, basically, this is the most immediate protection against blood loss. It is the prompt constriction of a broken vessel, and several things will trigger this reaction. An injury stimulates pain receptors, some of which directly innervate nearby blood vessels and cause them to then constrict. This effect lasts only a few minutes, but other mechanisms then take over by the time that it subsides. Injury to smooth muscle of the blood vessel itself causes a longer lasting vasoconstriction, and platelets release serotonin, which is a chemical vasoconstrictor. Thus, the vascular spasm is maintained long enough for the other two hemostatic mechanisms to come into play. The second one is the platelet plug formation. Platelets do not adhere to the endothelium that lines healthy blood vessels and the heart. The endothelium is normally very smooth and coated with prostaglandin, which is a platelet repellent. When a vessel is broken, though, the collagen fibers of its walls are exposed to the blood. Upon contact with collagen or other rough surfaces, platelets grow long, spiny pseudopods that adhere to the vessel and to the other platelets. The pseudopods then contract and draw the walls of the vessel together. The mass of platelets thus formed is called a platelet plug and may be able to reduce or stop minor bleeding. Here is a picture. So we have a break in the vessel wall. Blood starts to escape through this break. Platelets then adhere to one another and to the end of the broken vessel and to the exposed collagen. They then form a platelet plug, and this basically helps control the blood loss. The blood is no longer escaping through the break in the vessel wall. Then we have coagulation. Coagulation, which is the clotting of the blood, is the last but the most effective defense against bleeding. It is important for the blood to clot quickly when a vessel has broken, but equally important for it not to clot in the absence of vessel damage. Because of this delicate balance, coagulation is one of the most complex processes in the body. It involves over 30 chemical reactions. So, you have procoagulants, which promote clotting, and you have anticoagulants, which inhibit clotting. And they are normally predominant because in the normal situation within your body, you do not want your blood to form clots. You only want it to form clots when there is a problem, when there is a break. Perhaps clotting is best understood if we could first consider its goal. The objective is to convert the plasma protein fibrogen into fibrin, which is a sticky protein that adheres to the walls of a vessel. As blood cells and platelets arrive, they stick to the fibrin like insects in a spider web. The resulting mass of fibrin, blood cells, and platelets ideally seals the break in the blood vessel. The complexity of clotting lies in how the fibrin is formed. Due to a broken vessel, procoagulant activity begins the clotting process. The clot actually forms in about 3 to 6 minutes, and clots are restricted to the damaged site, where procoagulants usually predominate. Here are the steps. You have tissue damage. First thing, blood vessel spasm. then we form a platelet plug, and then we have a clotting mechanism, which is prothrombin activator, which deals with prothrombin and thrombin, which then deal with fibrogen and fibrin, and finally you have a blood clot formation. Once the clot begins to form, it launches a self-accelerating positive feedback process that seals off the damaged vessel. Most proagulins are proteins produced by the liver. They are always present in the plasma, but in an inactive form. But when one factor is activated, it functions as an enzyme that activates the next one in the pathway. That factor then activates the next, and so on and so on. And this sequence is called a reaction cascade. A series of reactions, each of which depends on the product of the preceding one. Many of the clotting factors are identified by Roman numerals, which indicate the order in which they were discovered, not the order of that they are within the reaction. After clot formation, fibrin contracts and pulls the damaged tissues closer together, and fibroblasts then migrate into the clot and repair the damage. There is also something called tissue plasminogen activator, TPA, which converts plasminogen into plasmin. Plasmin breaks down the fibrin and dissolves the blood clot at the end. Eventually, when tissue repair is completed and the clot must be disposed of, fibrinolysis is the dissolution of the clot, and this is achieved by a small cascade of reactions within a positive feedback component. In addition to promoting clotting, factor VIII catalyzes the formation of a plasma enzyme called kallikrein. Kallikrein in turn converts the inactive protein plasminogen into plasmin, which is a fibrin dissolving enzyme that breaks up the clot. Thrombin also activates plasmin, and plasmin indirectly promotes the formation of more kallikrein, thus completing a positive feedback loop. Now we are going to talk about human blood types. You may or may not know your blood type. You should know your blood type. Basically, there are 4 blood types: A, B, AB, and O. And then there is something called an Rh factor, and that is either a plus or a negative. So your blood type is determined by the specific antigen on the red blood cell plasma membrane. An antigen is a special protein. Antibodies in the blood will recognize antigens not normally found on the red blood cells of that person. Agglutination occurs when an antibodies bind to these foreign antigens, and they cause the foreign red blood cells to then clump together. When someone has a transfusion, that is a donation of whole blood into an individual. And it usually is done with the same blood type. But not always. Depending on what is available and what the situation is, the person may not be getting the exact same blood type that they have. So care must be taken to ensure that the blood that they are getting is going to be compatible with their body. Because if it is not, the body will recognize this new blood as a foreign invader and can cause widespread agglutination and even fatality. So, your blood group type depends on the presence of either antigen A or antigen B on your red blood cells. This gives us the AB, and O blood types that we see. Blood type will possess antibodies against the antigens not present on their red blood cells. So here is a picture. Column 1, antigen A, antibody B. That gives you type A blood. Second column has antigen B and anti-A antibody. That gives you type B. Type AB blood has both antigens A and B and neither antibody. Type O has neither antigen, but both anti-A and anti-B antibodies. If you have had your blood typed, it's a very simple process. So literally, it involves the application of anti-A and anti-B antibodies to drops of blood. If an agglutination occurs after an antibody is added, the antigen is present on the red blood cell. So you can even do this yourself in a lab setting. And you have maybe a card and you put a couple drops of blood down and then you add these antibody serums to it, and depending on where you see the clumping, you can read and determine your blood type. Here is a little picture of what is happening. So the first row, there is no agglutination. Type A blood is donated to a type A recipient, so no agglutination reaction happens. So at the end here, you see just 2 free red blood cells. The second situation is where we see some agglutination. So now we see type A blood donated to a type B recipient, which is causing an agglutination reaction because the anti-A antibodies in the recipient combine with the type A antigens on the red blood cells in the donated blood. So the last picture there is showing basically a clump. 2 red blood cells are now attached through the antibody, and this will cause clumping. This is the agglutination. This is what's detrimental. When transfusing blood, antigens in the donor and recipient must be compatible. This ensures that agglutination will not occur with the donated red blood cells. Type AB blood is considered to be a universal recipient. It possesses no antibodies, so it will not react with any donated red blood cells. So AB blood can get A, B, AB, or O. Type O is considered to be a universal donor. It possesses both antibodies and will react with all donated red blood cells except those of another type O. Then we have this Rh factor. And it's called the Rh factor because it was first found in a rhesus monkey. And it's the D antigen. If you have this antigen, you are Rh positive. If you do not have this antigen, you are Rh negative. The majority of people are Rh positive. If anti-Rh antibody is added to blood and agglutination occurs, the Rh antigen is present. So, people that are Rh positive can receive positive or negative blood. people that are Rh negative can only receive Rh negative blood. Rh negative persons do not normally have the anti-Rh antibody, meaning it's only formed, they only produce these anti-Rh antibodies after exposure. The first exposure will build the antibodies, but usually there is no agglutination. But with subsequent exposures, then agglutination will happen. So if you are Rh negative and you are exposed to Rh positive blood, the first time you probably will not see an effect. But the second time, there could be some trouble. Now, why would somebody be exposed to an Rh positive? If you are Rh negative, what situation might occur that would cause you to be exposed to Rh positive blood? Can anyone think of one? Well, it's pregnancy. So there's something called erythropoiesis fetalis. And this is a disorder in newborn infants resulting from destruction of fetal RBCs by maternal antibodies. If an Rh negative woman is pregnant for the first time and the fetus is Rh positive, nothing will really happen in relation to the fetus. But the fetal RBCs will enter the mother's bloodstream. So now the mother is starting to build up these anti-Rh antibodies. This is the first exposure that we just talked about. So nothing's going to happen to the fetus, and the mother is okay, but she now has these anti-Rh antibodies within her. Meaning that if she is then again exposed to Rh positive, we're going to see a reaction. And this is what happens. When the mother becomes pregnant then with a second fetus that is Rh positive, the mother's anti-Rh antibodies will pass into the fetus's bloodstream and result in agglutination of the fetus's RBCs. Does that make sense? So here you have two people conceive a child. The mother is Rh negative, and she is pregnant with an Rh positive fetus. It's okay. First pregnancy, not a problem. The mother has had some exposure to these Rh positives and has become sensitized to the antibodies, but nothing happened. Then she becomes pregnant again with an Rh positive fetus, and now the maternal antibodies actually attack the fetus's blood cells. Now, there are shots nowadays that can be given to females, to women that are pregnant, that are Rh negative. There's things that can be done to prevent problems. But it's important to know your blood type and your positive or negative sign because of things like this. Fetal red blood cell destruction reduces the oxygen carrying capacity of blood, and rapid red blood cell production releases immature red blood cells. And immature red blood cells cannot carry oxygen efficiently like a mature red blood cell. So this can have some detrimental effects. Freed hemoglobin can interfere with kidney function. Excess bilirubin can cause jaundice. And low oxygen and high bilirubin can cause brain damage. Afflicted infants can be treated by replacing their total blood volume with Rh negative blood. This then will add functional red blood cells and clear out that excess bilirubin. Now we're going to talk a little bit about some disorders of the blood. There are 3 groups: erythrocyte, leukocyte, and clotting. Erythrocyte disorders, the main one is anemia. It is the most common blood disorder, and it is basically a decreased oxygen carrying capacity in the blood. It is caused by a decrease in red blood cell count or inefficient hemoglobin. It can cause you to be weak, to feel lethargic, because you're literally not transporting oxygen like you should be. And there are several different types. So, nutritional anemia is an insufficient iron and vitamin B12 in diet. If it is caused by a hemorrhage, that is caused then by an excess loss of red blood cells through bleeding. So you're actually anemic due to loss of blood. Pernicious anemia is due to a deficiency of the intrinsic process. So if you don't have the correct intrinsic factor being released, you cannot absorb B12. And that deals with red blood cell production. There is also hemolytic, which is the premature or rupture of red blood cells, and aplastic anemia, which is due to destruction of red blood cell marrow or its inability to produce enough red blood cells. There is also something called sickle cell anemia, and this is due to an abnormal hemoglobin that cannot carry enough oxygen. So a red blood cell has that biconcave shape, which is suited very well for carrying oxygen. A sickle cell literally has a shape of a sickle, and that shape does not allow that cell to carry oxygen efficiently. And it also isn't pliable, and it can get caught and clogged in vessels. So people with sickle cell can have a lot of pain in their joints because of where these cells are pooling, and they usually suffer from fatigue, and it can be extreme. It's not good. Usually it is seen in people of African descent, and that is because it goes hand in hand with malaria. So malaria is a disease that affects the blood, and if you have sickle cell, you do not get malaria. So although sickle cell isn't good, it does prevent malaria, which is why we see it in Africa, because malaria is very prevalent in that tropical zone of Africa. We talked about the erythroplastosis fetalis already, and this has to do with the Rh positive and Rh negative factors in a fetus, and this can cause the fetus to have anemia, jaundice, or other problems. Polycythemia is if you have excessive numbers of red blood cells. So now you have too much, and this increases the blood volume and viscosity of the blood, which impairs circulation. And it could be due to cancer. Basically cancer of the red blood cell forming cells. So now they've just kind of gone out of control. They've jumped out of that cell cycle and they are overproducing. We have some leukocyte disorders. Infectious mononucleosis, best known as mono. This is a contagious disease of lymphoid tissue by the Epstein-Barr virus. We usually see it in young adults. Sometimes it's called the kissing disease, right? Because the common mode of transmission is kissing. It infects your B lymphocytes and it gives you a fever, sore throat. You can become very lethargic. And it can last a long time, as it says here. It can last years. So, not good. Leukemia, also not good. This is cancers of the cells that form the white blood cells in the bone marrow. So now we have excessive white blood cell production that inhibits red blood cell and platelet production. We can see acute forms in children and young adults, and we can see chronic forms in adults. Treatment could include chemotherapy and bone marrow transplants. Then there are some clotting disorders. Hemophilia. Hemophilia is a disease that we mostly see in males, and that has to do with the genetics and how it's inherited versus the X and Y chromosomes. It is an involved spontaneous bleeding and reduced ability to clot blood. So there is an actual lack of clotting factor. So we talked about that cascade a few slides back and how each factor makes more factor. So here you actually have a lack of these factors. So there's no clotting taking place and you can literally bleed out. Treatment involved can be a transfusion, which then would be basically containing these missing clotting factors because you're getting new blood with this factor in it. Thrombocytopenia is an extremely low platelet number that permits spontaneous bleeding to occur. And then we have thrombosis. This is a formation of a clot in an unbroken blood vessel. It may happen because an area has been roughed up or damaged or banged. Maybe you banged your knee. So I mentioned earlier that you want to be able to form a clot if you have a broken vessel, but you do not want to be forming clots if you don't have a broken blood vessel. And that's what this is talking about. Because if you do form a clot and there isn't a break in the vessel and that clot or embolus basically breaks free and gets into the bloodstream, it could move throughout your body and block something. It could block a vessel to your brain or your lungs. It could cause a stroke. It can cause death. So we do not want blood clots in places that do not contain a broken blood vessel. Remember, we said the anticoagulants are usually more found in higher numbers because for the most part you don't want your blood too thin, of course, anemia, but you want it at the right viscosity. You don't want blood clots just forming because obviously they can get into vessels and block things, and that can and usually does cause death. And that brings us to the end of our lecture. Please let me know if you have any questions. Hope you enjoyed it. Take care.